Thank you, Tom. It's a real pleasure to be here. I've been to the GoldLab Symposium several times, but this is my first time being invited to speak. I feel a little bit like a guy who's gone to a music festival many times and then his band gets invited to come play. But then he shows up and he finds out that he's on right after Eric Clapton and Jimi Hendrix. And not only that, but the MC is Paul McCartney. And to make matters even worse, his wife's band played the day before and blew the roof off. So, you know, it's a tough act to follow. But I'm going to try to stay in tune. So I'm talking about the operating system of the cell. What do I mean by that? But first of all, let me just, I know this is a diverse audience, a lot of varied background. So I like to, when I give talks to groups like this, to use advice from Max Delbruck, who said, assume your audience has zero knowledge but infinite intelligence. So what that means is I'm going to start off really basic stuff, and I apologize to you who know all of this stuff, but I'm going to try to move pretty rapidly up to the current stuff. So what do I mean by operating system of the cell? The easiest way to sort of think about the problem is think about the human body has all these different cell types. This is just a smattering of them. There are literally hundreds of different cell types, or thousands, depending on how you divide them up. They all have the same DNA, right? So they all have the potential to make exactly the same set of genes, but each cell type makes a subset of that. And so that's the whole issue of how you control the expression of genes. You can activate subsets depending on the function of that particular gene. So that's the problem, the biological problem I've been interested in my whole career, how you regulate gene expression. The strategy is to start with simple systems, bacteria, phage. You might say, well, bacteria, they don't have lots of different cell types. But they do gene regulation because of their environment. They turn on genes and turn off genes depending on where they are, what their environment is like. And this work, as Tom said, I was a grad student with Larry Gold, so that's where it started. And there was another grad student at the same time named Tom Schneider, and we did a lot of this stuff together. So I mentioned bacteria. So Sean and Rich both yesterday went back to the 50s and 60s. I'm going to do the same thing for the first regulatory system that was actually worked out and understood. So again, most of you know about this, but maybe some of you don't. So what I'm showing here, each bar is a little segment of the E. coli genome. In yellow are 4 different genes. The LAC-I gene makes the repressor, which is this red ball here, the LAC repressor. These other genes, those are required for the cell to utilize lactose, the sugar lactose. If there's no lactose in the environment, it would be a waste of energy and resources to make those proteins. So it doesn't. If there's no lactose around, the LAC repressor sits on the DNA at this O sequence, and it turns it off. Those genes simply aren't made. If there is lactose around, then the lactose binds to the LAC repressor, and it comes off of the DNA. And you start making, and this is the polymerase, which binds to the promoter. That's, every gene has to have a promoter. That's where the polymerase sits that's going to make the RNA, copy the DNA into RNA so you can make the proteins. So if you remove the LAC repressor, the polymerase can sit out the promoter and make this. But if there's glucose around, E. coli really prefers glucose. It's much more efficiently used, so it only makes a little bit of these enzymes. If there's no glucose around, then a small molecule called cyclic AMP is generated inside the cell, and it binds to this other protein, CRP, and then it binds to DNA, and it boosts the expression. Because if you only have lactose and no glucose, you want to make a lot of these enzymes because you're going to use the lactose to grow. So that's gene regulation, depending on the environment. Now, eukaryotic systems are much more complicated, but the general principles hold that there are proteins. Every protein has to have a promoter to start expressing stuff, and you can turn it on and turn it off with other kinds of proteins, generally referred to as transcription factors. Let's see. So when I first started grad school, my project was to sequence a little bit of the R2 gene that Sean mentioned yesterday. I was going to sequence about 100 base pairs probably, and we'd take 3 or 4 years. That's what it took other people. But 2 revolutions happened. while I was a grad student. The first one was they invented DNA sequencing. So now, that project that was going to take me 3 or 4 years was done by somebody else in a matter of a couple months. And so I had to find a new project, but that was great because now there's a whole new type of data, new types of questions you can ask. And furthermore, the other revolution that happened at the same time was the computer revolution. I mean, computers have been around for a while, but in 75, 76, suddenly everybody had access to computers. Microcomputers were being made. We had computers in our lab. My thesis was the first one done on a word processor instead of a typewriter. So that sort of stuff. So we had the opportunity to study what we wanted to study, but to use and generate our own data, but also take advantage of other people's data, because everybody was doing a lot of sequencing now. GenBank didn't exist in those days, so Tom and I would go to the literature where people would publish the sequence in the literature, and we typed it in. We had about 250 base pairs of sequence that we typed in from the literature, and then we would write programs to analyze it in various ways. So our goal was to understand how these proteins recognize certain sequences of DNA, or what sequences individual proteins recognize, thinking, well, then you could make predictions about what other genes might be regulated. You could make predictions about the effect of mutations. You could do synthetic biology and make new systems and so on. So the first thing we did is to set out to say, let's try to figure out what the sequences are that certain proteins want to bind to, that they like to bind. So I told you this CRP protein, in the absence of glucose, binds to DNA and turns on the next thing. So we said, well let's find out what that signal is. So this wasn't our work, somebody else sequenced this binding site for CRP, and we said, okay, that's one binding site. It probably doesn't need all of those bases. Let's sequence another one. Okay, so only some of the bases are conserved, left here in capital letters. So those are probably the important bases. Then someone seeked another one, less and less. It turns out, pretty soon, nothing's conserved. Every sequence, there's not a single position that every example contains. So our naive notion that there would be some particular sequence pattern that the protein recognized and bound to, that would be the key, and then we could use that to make all kinds of predictions. That was clearly false. But these sequences aren't random. And so what people would come up with is what they call a consensus sequence. You could take the sequences, line them up, and say, well, most of the time, you have a T in the first position, G there, and so on. These ends mean those positions are pretty much random, they don't matter, and so on. And now you could take this pattern and you could search the genome and predict which genes are regulated. But the problem is, if you search for this exact pattern, you get no hits. None of the known binding sites match the consensus exactly. So you have to allow a mismatch, and then you get some of them. You need 2 mismatches to get most of them. Some of them you need 3 mismatches or 4 mismatches, but the problem is if you start adding more and more mismatches, then you start getting lots and lots of false positives too. And in those days, if you went to the literature, you would find lots of papers describing what's the optimum way to use consensus sequences and allowing mismatches to do the balance between false positives and false negatives. We ended up coming up with a different idea, which was, I'm going to show a picture. Pictures were invented quite a bit later, but it's easy to describe our idea by looking at this picture. The idea is that each base contributes just abstractly, some number of points. And what separates the good binding sites from the non-binding sites is the good ones have more points, right? And so then the question just becomes, how do we find the right set of points for this particular protein that we can separate the ones that it binds to and the ones that it doesn't? And I initially did that. We went to computer science, we laid out the problem, how do we find a set of points that separates good ones from bad ones? And he said, well, have you ever heard of a perceptron, which is a simple neural network? It does exactly that. It does trial and error training. And so I built a little perceptron that succeeded in finding the set of points that would separate the good guys from the bad guys. Now, it turns out, before long, people realized, actually, there's a simpler way to do it. You mostly just count. You take the probability, oops, 1 second. I have the same issue that everybody had the other day. That you can actually just do this based on the probabilities of how often A, C, G, or T occur at different positions. And so Tom actually then took that a step further into information theory, and so the scale here is in bits of information. But the idea is pretty much the same. The idea is that underlying this, I'm showing you this picture, like I said, Tom invented this called the sequence logo many years later. In the early days, all we had was a matrix of numbers. And I think that contributed to the fact that even though we had come up with this method, and it was clearly demonstrably better than the consensus sequence methods, it didn't get adopted very widely until these pictures came out. And then people had sort of an intuitive notion of what those numbers in the matrix meant. And all of a sudden, everybody started using these pictures. If you go to a databases now, you just find them full of these kind of logo pictures. Okay, so there was another major technological advance a couple of years later, and that was the invention of DNA synthesis. And I would argue that's actually currently as important as DNA sequencing in molecular biology. I'm surprised he hasn't won a Nobel Prize. Anyway, it was invented by Marv Carruthers, who's here in the chemistry department. The building we're in is named after his wife. And he invented DNA synthesis methods. Now, nowadays, you can just go on the computer and you can order whatever DNA you want, and it'll show up in your mailbox the next day. In those days, that wasn't available. You could order the chemicals and you could make DNA yourself. We were thinking about, well, what sequences should we make to help fill in the gaps in our knowledge, because we still had limited amounts of examples of different things. And it occurred to us, and other people too around the same time, that we could make random DNA, or we could put random nucleotides. So these ends correspond to all possible bases at those positions. So we synthesized one thing and we had to get up 16 different sequences out of it. This one has 12 random positions. So that's 16 million different sequences. And we're not going to sequence all 16 million. We're just going to pick out a subset of those. In fact, in this experiment, I think we picked 180 colonies and sequenced them. But in addition, the protein that we were expressing here, this is a ribosome binding site, so it's the beginning of protein synthesis. That protein is easy to measure how much of it is made by a colorimetric assay, so we could have quantitative data. The range was from 1 to 3,000 fold changes in gene expression. And now you could, in fact, use a simple statistical method, linear regression, and find the parameters of your weight matrix that optimize the fit to the data. And in fact, not only would it give you the best fit, but you could ask how good is that fit. So in fact, with this case, it's 180 sequences, 3,000-fold range, our R-square, that is goodness of fit, was about 0.8. So not perfect, but not bad for a very simple mechanistic model. Okay, so the next challenge we had was, I showed you at the beginning that we had a bunch of binding sites for the CRP protein. Getting down to the actual binding site, the little piece of DNA that it binds to, that's hard, actually. It takes a lot of work. What's much easier, in fact, those pieces came because people had previously identified what genes are regulated by CRP, and then they narrowed down where the binding site was. Well, I thought, well, what if we knew that the set of genes, the light lines right here, these long lines, that's the upstream region of a set of genes that are all regulated by CRP. So there must be somewhere hidden there the CRP binding site. So can we discover it without actually narrowing it down to the thing, just by looking for the pattern that has maximal information content, which means most statistically significant. So here's an example of the actual data, sort of like the slide that Sean showed. Just a lot of A's and T's and G's and C's, but about 100 base pairs long. But the point is that we came up with an algorithm that would take that as input and generate that as output. One thing I found, this isn't true in all areas, but in the field of bioinformatics, one way that you can write a paper that gets a lot of citations is you come up with a new problem like motif discovery, you write an algorithm, and as soon as the computer scientists come across it, they'll write their own algorithm, and then they have to cite your paper when they describe why theirs is better. So I'm going to jump ahead 20 years. I'm skipping over, because all of that I just talked about there, that was all done in the 1980s. And then I'm going to come back to stuff that happened later with a new technology, which is what's sometimes called next generation sequencing. The 20-year interim, we weren't just sleeping. We were doing other things. We were using neural networks to do gene prediction. We were looking at RNA structure prediction, a whole bunch of other projects. But the advent of what's called next generation sequencing opened up a new line of research to get back to the problem of motif discovery. So let me describe what CellX is. This was invented by Larry. He did it. He would make random pools of RNA. You have a protein here that you mobilized in some way, and you just flow that over it, and the RNAs that bind well to the protein stick to this. You wash off the unbound stuff, and you wash off the bound stuff. But in the old days, you'd have to do this multiple times. You'd amplify, you'd do this, so you keep enriching for the guys that really bind well, and eventually you sequence, and you get, I don't know, 50, 100 sequences, whatever, to learn what it is that has a high affinity for your. Well, the advantage of the next generation sequencing is it's easy to get not only 1 million reads, but 100 million reads. And so what that means is we don't have to do the amplification step. We just take, and the other thing is we're doing DNA, not RNA, which is simpler because we don't have to worry about secondary structure. It's just a linear piece of information. So we made random pools of, come on back. Random pools of DNA, separate the ones that bind and ones that don't, and then just sequence the bound ones. And the idea is that the higher probability of binding corresponds to better binding energy. And so actually we want the quantitative data. We don't want to be enriched for just the best sequences. We want to get the whole range of quantitative data from the best to the medium to the low binders. And you can just take that data and plug it into some programs and end up with a logo like this. But actually in our case, I'm showing you a logo of information content, but actually underlying that is actually now estimates of the actual binding energy contributions of each base at each position. Now we did a little bit of this. Ours was sort of proof of principle. I didn't really ever want to get into the big mass data generation field, but other people did. So we did some collaborations with Scott Wolf, mostly on zinc finger proteins, which were the earlier version of CRISPR, we're doing gene editing. Martha Bullock had her own method for doing it. Tim Hughes collaborated with lots of people. But I want to emphasize UC Topoli, who was in Sweden at the time. So he came up with essentially the high CellX method that I talked about. He called it multiplex high throughput CellX. And a month after our paper came out, he came out with one that had 20 different transcription factors in it. A couple of years later, 240 transcription factors. So what he had done is he'd taken this CellX approach, which is pretty straightforward, and automated it. He had robots do all the work. And so a year after that, he had 1,000 different transcription factors. Okay? There's about 1,500 transcription factors in the human genome, and now, given what everybody's been working on, we know the motifs for almost all of them. There are some that are still problematic, but we know the most. We know a lot. Okay, I'm getting close to end of time. I'm going to jump ahead. So how do you use motifs? I'll just show you one application, or I'll just describe one. So there's a whole field called genome-wide association studies, where you sequence, if you have sequences of lots of people, you can compare their phenotypes, whether they have their skin color, their hair color, all kinds of stuff, their height, and you can identify variations in the DNA that correlate or associate with those things. You can also look for events where people with a particular disease have particular mutations in their DNA. So the vast majority of GWAS hits are in non-coding. Maybe not surprising, most of the DNA is non-coding. But you always hear about, well, somebody found a mutation in a gene that gives you a high risk of Alzheimer's or high risk of this. But most of the variants, whether they're for normal phenotype variation or disease, are in non-coding regions. So it suggests that what they're doing is they're altering expression, not the function of the genes themselves. I'm just going to show one example because it's a sort of an obvious, easy one to do. So the gene here is this. The gene here is, come on, back up. Is TERT. It's part of the gene that for making telomeres at the end of chromosomes. So normally when cells divide, their telomeres, which are the end of the chromosomes, get shorter, and eventually they can't divide anymore because the telomeres are too short. But if you can turn on the enzymes for lengthening them, the cells will continue to divide. So cancer cells, the characteristic of cancer cells is they keep dividing forever. And so there's usually some mutation that initiates the cancer, but it'll just stop dividing if you don't do something about the telomeres. So the most common non-coding variants in cancer, out of all the cancer genes, you count how many times they are these mutations in the TERT promoter right here that create binding sites for transcription factor, the GABP transcription factor. Okay, so it occurred to me, so the thing I showed you about using CellX to identify things, there were a bunch of assumptions. We had to assume that the data was generated by maybe a Boltzmann distribution or a Fermi, some kind of distribution. We also are making the assumption that the positions are all additive, that's a common assumption. It's usually not a bad assumption, but it's certainly not perfect. It occurred to me we don't have to do that. So I'm going to describe a method we came up with called SpecSeq for specificity by sequencing. So this is my only equation, I think. So this is just the reaction. We have a protein and some sequence I in a test tube, and it goes together, it makes a complex, and it comes apart. It's in equilibrium. And you measure what we call the association constant for that particular sequence by just measuring the concentration. These brackets here represent the concentration of the complex divided by the free concentration of the two components. That's the association, and the higher the association, the better the binder. Okay? So one way to separate the bound fraction, the DNA that's bound to the protein from the DNA that's not bound to the protein, is you can what's called run a gel. Here's the test tube, the protein and the DNA, run a gel, and you put an electric current that pulls the DNA through. If the DNA is bound to the protein, it goes slower than if it's unbound. So I could just cut out these bands, but I'd have to know what the protein concentration is to know the affinity. But if all I care about is the relative affinity, the protein's concentration goes away. Sorry, I lied. There's another equation here. The relative affinity of sequence 1, 2, 3, and 4, all of that information is contained in those two bands. I don't have to make any assumptions about additivity. I don't have to make any assumptions about is it a Boltzmann distribution or not. I could just make a list of every sequence in my library and say this one is 2 times better than that one, it's 4 times better than that one, etc. So we wanted to test this out. And remember I mentioned the lac repressor. So the lac repressor has always been a bit of an enigma. It was this first regulatory protein that was described. It was never, well, it was crystallized, but not good enough quality that actually anybody ever got a good structure from crystallography. It has an asymmetric pattern, even though we knew it bound as the dimer. And there were a lot of questions about it. Somebody else, Jack Stadler, actually had deleted the middle base here and then changed the right half to look like the left half, so make it symmetric, and it actually bound a little bit better. But nobody had ever done a really comprehensive analysis, so what we did was we deleted the middle base and then we randomized the asymmetric positions. We also added a new base in the middle and randomized the asymmetric positions. And we also left the middle base, randomized it, and randomized the left half or the right half, and we just said, what are the best possible sequences? But partly I want to highlight this graph at the bottom. So this is 2 different experiments, sample 1, sample 2. When we submitted this for publication, one of the reviewers said, I don't want to just see a technical replicate, I want you to do a new experiment. Well, this is actually 2 different experiments at 2 different protein concentrations. I told you the protein concentration doesn't matter, right? It drops out of the equation. Well, this verifies it. And in fact, the variance you get is about 0.1 kT in terms of energy. So I think this is the most accurate high throughput method there is for determining things. And like I said, we haven't invoked any particular model of additivity or anything like that. So what did we learn about? So if you delete a base and you randomize it, you get a symmetric pattern. If you add a base, you also get a symmetric pattern, but they're different patterns. This is the structure from Rob Kaptein's lab determined by NMR, and it shows that you have what we call a compressed and an extended version of the protein. Protein can bind DNA in 2 ways, but importantly, those 2 ways lead to different motifs. So the wild type is a mix of the compressed motif up here and the extended motif right there. So interesting sidelight. Here's the actual, so now I've changed logos. I'm now showing you directly the energy. We've set the average to 0, and the good guys are on top, so it's actually minus energy. But this shows you the change in energy for all possible changes to the sequence. It then occurred to me that, a lot of proteins bind cooperatively. What that means is, so suppose I have a binding site that has binding for 2 proteins, X and Y. Protein X binds alone, it goes over to here. So that would be the association content for protein X. Or protein Y binds alone, so this would be the association constant for protein Y. But the question is, well, how does X binding compare if Y is already bound? So I compare this number to this number. If they're the same, it says they're binding independently. They don't care about the other one. This number could go to 0 if binding of Y prohibits binding of X. But the more interesting case is if this number is bigger than that number. So that means that binding Y increases the probability of X binding. And again, you just run a gel and you get all the parameters you want. Not only the specificity of the different things, but the cooperativity values. There's a bunch of equations here, but I'm just going to skip over them. I just described what you can learn by simply sequencing bands in a gel. We tested this, so two proteins, SOX2 and OCT4, were known to be important in a whole bunch of developmental things. They knew that they both worked together. It wasn't clear how whether they're cooperative or not. This guy, Ralph Yauch in China, had been working on it, and I wrote him and said, we can do this thing in parallel. Instead of doing one at a time, let's do hundreds at a time. So my student, Kenny Chang, went to his lab. We took the two proteins with binding sites for SOX and OCT. Damn. And we put them in all possible orientations, and we varied the spacing and stuff like that. And just to jump to the next thing, we found out that, yeah, it does bind cooperatively, but only in one orientation and only at a certain spacing. So that was interesting information that hadn't existed before. So now I got to actually apply this to a real medical problem. I'm mostly just a basic scientist, and I'm happy when people use my methods, but this was an example where Shiming Chen is a professor in the ophthalmology department, and Yichao Zhang was her graduate student. She's now a postdoc at MIT. But they were studying a protein called CRX, which stands for cone rod homeobox gene. It was a transcription factor that was known to be essential for development of both cones and rods in the retina. And there's a whole bunch of mutations in it, and they've been studying it for years. Yichao was specifically interested in two mutations that were in the binding domain, the homeobox binding domain of the protein. One of them changes glutamate to alanine at position 80. Another one changes a lysine to an asparagine at position 88. So the first thing we did was we measured what she did, was measure whether there's a change in the specificity. And you can see changing this lysine to asparagine changes the specificity entirely. So that one's easy to understand. The protein doesn't turn on the genes it's supposed to. It turns on a bunch of other genes instead. So that's clear why that one is a mutant. This one is not so clear. The motif doesn't really change. If you look at the scale, it's a little less specific. That is, it's a little more likely to bind other sequences. But it doesn't change very much. So it wasn't clear why that one was leading to disease. And it turns out, I'm not going to show you all the data, but what Yichao did was measure the cooperativity. And it turns out that the wild type protein, so a lot of the promoters for these photoreceptor genes, some of them have just one binding site. Here's the consensus sequence. Some of them have a single site plus a site for a dimer, where it binds head to head. This has a lower affinity. It doesn't match the consensus very well, so it doesn't bind at low concentrations. But what happens is, because it binds cooperatively as the protein concentration goes up, here's showing what's happening at the motif activity. For the good, strong binding sites, it comes up pretty quickly, and then it sort of levels out. For ones that are dimeric, it comes up later because it requires more protein concentration, but it ends up at a much higher level. And so what happens is that in the wild type cell, you turn on a set of early genes, and then later you turn on a set of later genes that have higher expression, and this mutant never turns on those later genes, or it doesn't upregulate them. And so that explains why that's a mutant in photoreceptors. All right, I have just a couple minutes. I want to jump to one final thing. So I've been talking about transcriptional regulation. It's ubiquitous, it's well understood. There's still a lot we don't know, but it's pretty well understood. But regulation can happen at all different stages of protein synthesis. Transcription termination, RNA splicing. Thank Tom for giving an introduction to that. Every one of these steps can be regulated and affect how what the final product is. I was particularly interested, partly because of stuff I did in Larry's lab a long, long time ago about genes that are autoregulated. Think about the cruise control in your car, right? You set it to some, and if you go up a hill, it accelerates so that it keeps the speed constant and all that kind of stuff. Cells can do the same kind of thing. They can set the level that they want of a particular protein, and if they make too much, they turn themselves off. And in bacteria, all the ribosomal proteins are autoregulated. So I figured that was likely to be true in yeast. There were like 4 or 5 examples that were known. Well, so what we did was we came up with a strategy where we did the following. There's a library, essentially, well, Erin O'Shea tried to make a GFP tag, that is, you add this fluorescent tag onto every protein. She got about 2/3 of them. So there are 95 ribosomal proteins that have a GFP tag on them. And the assay we did was just simple. We took a gene X on a plasmid so we could induce it. This is a promoter that you can turn on at will. You turn it and you ask what happens to GFP. If GFP goes down, then that means that there's some kind of feedback regulation. It doesn't tell you the mechanism. It could be transcription, it could be translation, it could be many different things. We found that 1/3 of the ribosomal genes had feedback regulation. Like I said, only 4 or 5 of those had been known previously. There were 30 out of our 95 had feedback regulation. I was specifically interested in ones. The first one we studied was just works like bacteria in that it binds to the ribosome binding site and prevents translation. So that's kind of boring because it's just like bacteria. So I was specifically interested in ones that might regulate via splicing. So I'm not going to show you the data, I'm just going to jump to the conclusion. So this gene is RPL7B. There is a structure. So here's the intron, the 5' end, the 3' end. Those have signals that lead to the spliceosome to cutting out that piece of RNA, as Tom described. There's another important signal here, the branch point site. So those 3 things are important. The intron itself is long, but there's this secondary structure that we refer to as an enhancer. Now, Michael Rosbash first demonstrated RNA enhancers in splicing in yeast in 1995, I think it was, right? 30 years ago, he doesn't remember. But that was an example that sort of led us farther down this path. There have been recent paper by the Dosh lab in Stanford where they did in vivo structure mapping, and they found that almost every ribosomal gene has what they call the zipper stem that brings the 5' end and the 3' end closer together. Well, the interesting thing about this is, so that's a structure, and we showed that it occurs if you mutate this so that it doesn't form this, then splicing goes way down. So that enhancer is required, and part of the reason it's enhancer is because of this branch point site. Unlike 90% of branch points in yeast or the consensus sequence, this is a mismatch to the consensus. If you make that a consensus branch point site, then the enhancer doesn't matter anymore. So the reason you need the enhancer is because you have a non-consensus branch point site. Now, this intron had been analyzed by different groups, one of which was Sean's wife, Elena Rivas, looking for conserved structures by comparing across a whole bunch of yeast. And there's a different structure than this one that they found that was conserved across a bunch of yeast genes. So it turns out that what happens, the way you do autoregulation, is this is a ribosomal protein, so its normal function is to bind to ribosomal RNA, right? So it's an RNA binding protein to start with. And what happens is this structure here, this alternative structure, so instead of this yellow binding to the red, it binds to the orange. That's the structure that is stabilized by the protein. So what happens is now when you make more protein than you need, it gets rid of the enhancer. It doesn't actually block it, because if you change this branch point to the consensus, it still splices perfectly fine. So it's only the fact that you've eliminated the enhancer that you've done autoregulation. And over time, I'm going to stop right there. I'm just going to put up this last slide of people who have been in my lab in various phases or people I've collaborated with over the years. Thank you for your attention. I'll be happy to answer some questions.
Thank you, Gary. So let's start out with raising hands and passing the mic around. Yes, over there.
Yeah.
Long ago, there was a lot of talk about junk DNA, which I assume they meant non-coding DNA, and they figured out that a lot of that had to do with gene expression. Do we have a sense for what percentage of that is involved in gene expression and how much we just still functionally don't understand at all?
Yeah, so it's an interesting topic. So junk DNA mostly meant repetitive elements that occur over and over again, and there's a whole class of them. And about 50% of the genome is these repetitive elements. There's also non-coding RNA, which is not part of repetitive elements, and people mostly think that's where most of the regulatory stuff happens. But now it's pretty clear that the repetitive elements are expressed in some, or they contribute to expression in some cases. Oftentimes in cancer they get activated and so on. So there's another quote from Sidney Brenner talking about junk DNA. He said, we call it junk DNA, not garbage DNA. Garbage is what you put out at the curb, it get hauled away. Junk you put up in your attic because you never know where you're going to need it, right? And so junk DNA is sort of this thing, we don't really know what it's for. It's now clear that it's sometimes useful or sometimes deleterious, but it's there and it's sometimes expressed, but probably most of the time not.
Yes.
Well, I'll let you call.
Are you able to tell, so I'm part of the Human Virome program right now. Are you able to tell which one of these might be viral in their origin, so like transposable elements and those kinds of things?
I think they're thought to all be initially from a virus, and then they get in and they jump around. But I think they were all of viral origin initially. I mean, you can trace their lineage. So humans and primates in general have a bunch that don't occur in mouse, for instance. Sean's a much more of an expert than me on this. One of his students actually did a lot of computational work on these families. But they all entered the genome at various times in evolutionary history. And I think they all originated as viruses.
Mary Ellen?
So I love these cool RNA tricks. They're really fun, where you bind a protein to an RNA and then change it. What I'm really curious about is your use of the word enhancer there. So what I typically think of as an enhancer is a DNA-encoded element.
Right.
And so would you include RNA-encoded elements as enhancers in these cases?
Well, ask Michael. He's the one who came up with the term, but it makes sense in this case. What it is, it's a piece of sequence, and usually you're right, the enhancer is usually refers to a piece of DNA that can be a long ways away from a promoter, but when factors bind there, they can turn on the expression of a gene, which might be far away. But that's the fact is that something binds to it and it increases. So in this case, the enhancer is the structure itself because it increases the expression. And I think it's a valid use of the word. It's a different use than has been, but like I said, I rely on Michael Rosbach's use of it from 30 years ago. So, and I think it's a valid, because it describes what that structure does. It enhances splicing, and it's actually necessary because of the non-consensus branch point site.
In the case of the splicing enhancer being tied up by the ribosomal protein binding to that element, how close does the structure of that RNA in the intron look like to the piece of the ribosomal RNA to which that protein normally binds?
Yeah, we tried to do an alignment. The problem is RPL7 is a very elongated protein. And if you map where it binds on ribosomal RNA, it binds to like 3 or 4 different helices, little pieces of them. And we haven't been able to do a direct mapping over this. But this is also a complicated structure. And so somehow it mimics the site in some way or other. I wanted to make one more point that I forgot. It was actually came up in a conversation with Sean last night. So people who do RNA structure prediction, that includes me and Sean and almost everybody, I asked him if he knew examples, look for the structure. But RNA, more so than protein, easily exists in multiple different structures. And so what we ought to have are programs that look for not only the structure, the lowest free energy or the most conserved or whatever, but alternatives of that structure that might be involved in alternative functions, maybe regulation, stuff like that. And I don't know of anybody who's doing programming. He said he might know somebody, but in general, people don't do that when they look for RNA structure prediction.
Well, that's becoming more and more popular. So Sylvie Ruskin at Harvard is a leader in that area.
Okay, yeah. Okay, that's great to know. I didn't know that.
Okay, thank you, Gary.